In this project, we are going to perform a molecular docking process using HIV-1 Protease enzyme using AutoDock Vina. So the main objective of this project is to try and understand a complete docking workflow from the protein and ligand preparation to the docking process and interpretation of the results. So what we are going to do is we are first going to download the protein structure from the protein data bank and the ligand from PubChem, and we are going to prepare both molecules for docking. So we are going to first download the protein PDB information. So let's go to browser and then click PDB. Okay, and click. Okay, so we are going to type here. Okay, there it comes. So we can find information about the protein, like the structure. We can preview the structure and verify. Okay, then we can also find other information relevant to the protein. Okay, but the main goal for this is to download the files that we are going to use in the docking process. So we go to the download files, and then we are going to create a new folder which is going to act as our working directory. So let's go in and click and create a file. Okay, so the next thing we are going to do is to find the ligand information. So we are heading on to PubChem, and then we are going to search for the ligand Indinavir. So we go on and click download, and then we are going to download 3D conformer SDF file. That is the one we are going to use. So let's go on and click save. Okay, we are going to save in the same working directory. Okay, so now we have our two files, the protein information as well as the ligand information. So the next step is to prepare the protein and the ligand for the docking process. So let's go to the next step. So the next step in the docking process is protein preparation. This is where we are going to analyze the structure of the protein, and we are going to remove water molecules, remove native ligands, add some hydrogen atoms and charges. For proteins we add Coleman charges, and then we are saving the file is PDBQT. Okay, so since we have the PDB information that we have downloaded before, we are going to head on and visualize the protein in PyMOL and see if it has any native ligands that are already there, and probably we are going to get rid of them because they might affect the docking process. We can change the color as well to see clearly. So we are going to look for the ligand. As you can see, we have a native ligand inside, so we are heading on and click it, and then we are going to delete it from the parent structure. Okay, so for this process we can use the PyMOL command interface. We're just going to click the ligand, and then we head on to the command interface, and then we write remove selected, and then it is going to be removed. Okay, and then we are going to save the protein molecule in PDB format. Okay, then we're going to save the PDB format in the same working directory as before. Okay, so just label it as protein PDB. Okay, then we hit save. Okay, and then now we are going to open our ADT tools, that is AutoDockTools, and then we are going to perform the rest of the process for the protein preparation. Okay, so this is the AutoDockTools interface. Just going and load the molecule. We click read molecule, and then we'll go to our working directory. Okay, and then we take the cleaned protein PDB structure. Okay, and then this is the visual confirmation of the protein. As you can see, we have the meshy structure of the 3D protein. You can change as well the visuals. You can choose which one you are suitable to work on with. I'm more comfortable using the original meshy structure. Okay, so now according to the steps, we are going to first remove water molecules from the structure. So we're adding edit and then delete water, and then the next step is to add hydrogens. So we click edit and then you add polar hydrogens. For the protein, you're going to add polar hydrogens. Okay, and you can visually see that there are some structures which have been added on the 3D structure. Okay, and then the next step is to add charges. So you hit on edit and then go to charges. You add common charges. So simply we add common charges because they will provide a physically accurate representation of the molecule's electrostatic field. The last step is to merge polar hydrogens. So we're just going to simply click and then we merge polar hydrogens. Okay, so the last step is to save the PDB-QT format. Okay, so the next step is to prepare the ligand, pretty much the same way we have done for the protein. We're just going to use AutoDockTools. We're also going to add polar hydrogen atoms, assign charges, but this time we are going to assign Geistige charges, and then we're also going to define rotatable bonds, and finally going to save the PDB-QT file. So now that we have the SDF file, we're just going to open it and verify the structure of the ligand. So this is the 3D structure of the ligand. So we are just going to save it as a PDB format so that it is going to work well in AutoDock environment. So let's just go on and save. Okay, so the next thing to do is to open AutoDockTools and load the molecule. So let's just go on and open AutoDockTools. Okay, so we are going to load the ligand molecule. So this is the structure of the ligand as we have visualized in PyMOL. You can also change how you visualize the ligand. So now we are going to add hydrogens. So just go and click all hydrogens and then okay. And then the next step is to add charges. So we're going to compute case taker charges. Click OK. Now the last part is to assign some rotatable bonds. Okay, so we select the molecule. Choose torsion. So basically it is going to show you how many rotatable bonds and non-rotatable bonds are there. They are shown in different colors: green, magenta, and red. Okay, so basically it's just showing us during docking process which bonds can rotate so that they can securely fit in the pockets. So we are just going to hit on save, and then we are going to save a PDB QT of the ligand. If you have ever done a docking process, you might probably know that defining the grid box is one of the crucial steps. But well, in this case, I'm just going to show you how we define a grid box, but I might probably not use it because once I know the configuration that covers our binding pocket that we are targeting, I might just create a configuration text file, but I'm just going to show you how we define a grid box. So we are initially going to open the original PDB file of the protein in PyMOL. And then I'm going to try and locate the correct coordinates where the native ligand is already located inside the protein. So I'm going to use PyMOL's command interface to actually try and locate the center of mass, that is the correct coordinates of the ligand. So I'm just going to write center of mass, and it is going to show us the X, Y, and Z coordinates. So that will be our coordinates for the grid box. So once we know these values, we're going to simply open our AutoDockTools, and then we are going to and then we are going to load the molecule, that is the original protein molecule as well. Okay, and then after that we are going to define the grid box. So just click here, grid. Okay, open grid box. Okay, so since we now know what are the X and Y and Z center coordinates of the grid box, we're just going to simply place the values. So for X it's 13.043, and then Y 22.460, and then Z it was 5.596. okay, and then we are going to save. So as you can see, the grid box is now lying inside the structure, meaning that this is the location of our binding pocket. Okay, just going to save. Okay, and then we are going to save the GPF file inside our working directory. Okay, okay, so now that we have our target, which is the protein, then we also have our ligand prepared and ready, we are now going to perform the last step for this docking process, which is using AutoDock Vina to actually predict the binding of Indinavir and the HIV-1 Protease. So what we are going to do, as I have told earlier, I'm not going to use the grid box. Actually, I'm going to create a config file that is going to handle all the parameters. Okay, so I'm going to use Notepad to create the config file. Okay, so I'm going to define my receptor, protein PDB-QT. Okay, and then the next is the ligand, which I have labeled as ligand.pdbqt. So let's write here. Okay, so the next thing we are going to do is to define our grid box. Since we already have the values of x, y, and z, so we are just going to put the sender of x, sender of y, and sender of z. So let's do it. Okay, so we are also going to define the size of the pocket. So roughly the standard size that we can use here is 20. You need to cross check with the protein. If you ever need to calculate the size of the protein, you can use some tools like CastP to determine what is the size and the dimensions of your pocket. But for this case, I'm just going to use 20. Okay, and then we're also going to define what is the exhaustiveness. This is something to do with the type of PC that I'm using, and also the number of modes. And then lastly, the energy range. Let's just put 3. Okay, so after we have completed, let's save our file as config.txt. Okay, so since now we have everything, I'm just going to open command prompt, and then I have AutoDock Vina installed in the same working directory. So I'm just going to test if the AutoDock Vina is working by just typing vina.exe help. Let's just see if everything is working. Okay, as you can see, all the parameters has been loaded. Okay, so I'm going on to run the Vina config text, and as you can see in the background, it's now computing the docking process. So we're just going to wait until the bar reaches 100. Okay, so now we've got our results. As you can see, we have 9 modes from mode 1 to mode 9. They've been arranged in the order of the most negative affinity energy, which is the most stable, to the least stable. Okay, and as you can see also, it is showing us the distance from the root mean square value. Basically, it's interpreting that how far the binding is from the actual parameters that we have given. Okay, so as you can see, mode 1 is the most accurate one, followed by mode 2 up to mode 9. Okay, so after this, you can perform other various experiments using the data from docking. Like for example, you can do MD simulations. You can calculate that is the quantitative structure activity relationship. As well, you can perform, you can find all other drug like characteristics of the possible drug target. Okay, so that's it. Thank you.